and welcome to my poster presentation. Today I'll be discussing the 1KSA project and its role in advancing biodiversity genomics in South Africa. As you might know, South Africa is one of the most biodiverse countries in the world. However, a significant amount of genetic research on South African species has historically been conducted abroad due to cost and resource limitations. This has led to a drain of skills, data, and opportunities from the country. To counter this challenge and build local capacity for biodiversity genomics, 1KSA, a South African biodiversity genomics program, was launched in 2023. This initiative is supported by Diplomics, a research infrastructure program based in South Africa. 1KSA aims to support genomic research by offering sequencing services and making data available through a collaborative platform. The 1KSA workflow on the top right corner of the poster consists of several steps, starting with Step 1, Species Motivation and Application. Researchers submit an application justifying why a species should be sequenced and to see if it adheres to certain criteria. Step 2 is Sample Submission. Approved projects submit high-quality DNA samples to the lab. Step 3, DNA quality control and library preparation. The DNA undergoes quality checks and preparation for sequencing. Step 4, whole genome sequencing. We use Oxford Nanopore's Promethean platform for long-read sequencing. The sequencing and data analysis are provided free of charge to researchers, allowing them to focus on using the genomic data for their studies. Step 5 is draft genome assembly. Reads are assembled into a draft genome. Step 6 is data curation and storage. The assembled genome is stored and processed for quality control. Step 7 is data sharing and access. The final data is made available to the researcher or sample contributor at no cost to support further research and conservation efforts. The data management plan is available via the 1KSA website. Now that we have the sequencing data, the next step is assembling the genome. You can follow the process in the genome assembly pipeline workflow shown in the middle left of the poster. We use a Nextflow pipeline that runs on the Center for High Performance Computing Server, or the CHPC. This process involves several key steps, which include quality control, filtering out low-quality reads, genome assembly, reconstructing the genome using the Fly assembler, polishing and error correction, refining the assembly to improve accuracy, and then quality assessment. We look at several different metrics such as contig N50, number of contigs, average read quality, and BUSCO scores to evaluate completeness. To date, we have successfully assembled 77 genomes, covering a diverse range of species, including insects, fungi, mammals, plants, and fish. A key measure of assembly quality is BUSCO completeness, which checks how many essential genes are present in the genome. Our assemblies consistently achieve high BUSCO scores, with many exceeding 95% completeness as seen in the figure in the middle right of the poster. For this presentation, we are focusing on 4 representative species from our collection. You can see these species cards on the poster. Each card provides important details about the species' genetic data, including genome size, assembly statistics, and the IUCN conservation status. Additionally, all assembly statistics for each species are visualized in the snail plots, providing a comprehensive overview of genome assembly metrics in an intuitive format. Let's take a closer look at one of the examples, the common fierce ant on the bottom left of the poster. This species, belonging to the phylum Arthropoda, is a small but resilient insect known for its aggressive foraging behavior and widespread distribution. Understanding its genome provides valuable insights into social structure, adaptation, and evolutionary biology. You can see on the card that the estimated genome size of the common fierce ant is 259 million DNA base pairs, or rounding it up to 260 megabases. The sequencing run using the Promethean technology produced 30.93 gigabases of data with an N50 of 1.74 kilobases. The draft genome assembly spans 231.27 megabases and has a BUSCO completeness score of 98.1%, indicating a highly complete and high-quality assembly suitable for further genetic analysis. We also included additional species cards for other species of interest such as the Brodus whale, birds of paradise, or more commonly known as the Strelitzia, and a white steenbras. These cards provide genome assembly statistics and help highlight the importance of genomic data in biodiversity conservation. I hope this presentation gives you a sense of how 1KSA is contributing to South Africa's ability to conduct cutting-edge biodiversity research. By sequencing the genomes of a wide variety of species, we are paving the way for important conservation efforts, population genomic studies, and the development of bioeconomy solutions. Thank you for your time. If you have any questions, feel free to reach out. For more information, you can go visit the 1KSA website, and the link to our GitHub page is also available on the poster via the QR code.